Okay, so this lecture goes with Chapter 12, Evidence for Evolution. We'll be discussing some of the points of evidence that support the theories of evolution. So the first line is paleontology, which is the study of old geology and fossil records. What I mean by old geology is the historical patterns of geology of the Earth. And this is the first observed evidence for evolution, was in the fossil record. New organisms were being discovered in the fossil record than ever existed in the past. And this shook everything up because at the time people thought that the animals that existed now were the only animals that ever existed, and that made sense when you consider those are the only animals you ever experienced. So we had no idea that there were not only animals that used to exist that no longer exist, but there are whole assemblages of organisms that used to exist. We also didn't know that the organisms that existed today never existed in the past. So this all plays factors into it. The geological time scale also played a big factor of evidence for evolution, as we discussed in the previous chapter. James Hutton and Charles Lyell ended up giving the time necessary for evolution to occur. Later on, through radioactive dating, we were able to determine the Earth's age to be 4.6 billion years, give or take a couple hundred million years here or there. But it's certain now that it's in the billion-year range, not the million-year range, not the thousand-year range. Well, the interesting thing about it is, as we looked more into the paleontological record of living organisms, we discovered that time periods are marked by mass extinction events, and this is basically how we basically organized the different eras of time. It was based upon organisms that existed and organisms that didn't exist. And two particular big mass extinction events, the one being the most famous one, 65 million years ago, non-avian dinosaurs went extinct. For most of us when we were growing up, we're told dinosaurs went extinct 65 million years ago, but there's a direct link between dinosaurs and birds. And to some biologists, birds are considered to be dinosaurs, and to some degree they are. The same thing is we're apes and we're monkeys. Essentially is what we are. If you go follow the fossil record back far enough. I'm not trying to denounce us. We are humans, but if you go far enough back in the record, that's what we were. But despite the massive effect that this event had, it led to mammals and birds basically dominating the Earth from a macroorganism perspective. It wasn't nearly as big as an extinction event that occurred 248 million years ago, that opened up what we call the Mesozoic, went from the Paleozoic to the Mesozoic. 248 million years ago, nearly all of life went extinct, 90 to 97% of life. We're still working out what happened there. But then again, when I was growing up, I was often told we would never discover what ended the lives of the dinosaurs. We're pretty confident it was an asteroid or comet strike that around the Yucatan area of Mexico, 65 million years ago. But 248 million years ago, nearly all life went extinct. And these are sort of the barriers, the markers. And actually, a group of organisms called the trilobites went extinct 248 million years ago. And they were around for roughly about 300 million years. They dominated the oceans in terms of being like sort of a underwater cockroach. They went extinct. There's no other organism that we know of that has as much diversity in the fossil record than the trilobites do. Matter of fact, they discovered a 3-mile stretch up in upstate New York of a fossilized group of trilobites that were all mating. 3 miles of organisms mating with each other. What probably happened is they got hit by an underwater mudslide at one point, which basically suffocated them. And often what happens during these events, it's anoxic, so there's not much bacteria down there that can break down organisms in anoxic conditions. Anoxic means no oxygen, and the organism fossilized. So fossilizations occur by many different methods, but it's a rare process that occurs. It doesn't happen very frequently, doesn't happen that often, and we're lucky to have the fossil record that we do have. It's often noted that the fossil record is sketchy and has breaks in it and missing links in it, but that's to be expected because it occurs so rarely. So let's go over some of the ways that fossilization occurs. One is compression. This involves sediment below and on top of organism, results in the remains being left behind. The organisms are sort of squished inside the sedimentary layers. Petrification, on the other hand, results from the hard body portions of the organism, such as the skeletal structure of tetrapods. Tetrapods include amphibians, reptiles, birds, mammals, dinosaurs, all organisms that basically have 4 limbs, and some of them are missing, like snakes, but we all have 4 limbs. And fish, for that matter, also, they're not tetrapods, but they do have hard skeletons. So when petrification occurs, those hard parts are replaced by the mineralization of the soil that's around them, the sediment that's around them. This also will occur with like the shells of mollusks or the exoskeletons of arthropods. Now, petrification also occurs in plants as well. You might have bought petrified wood growing up. You might have a piece lying around. That occurs with wood. Wood will petrify as well, and it makes this beautiful brown sort of glassy stone-like structure. Imprints are another thing. Imprints leave an outline or an imprint of the organism in the rock. And this is a common way that leaves become fossilized. There's some beautiful fossilizations of, say, fern fronds, things of that nature. We have the imprint of the structures of the leaf left in the fossil record. To let you know, petrification, to go back to petrification, not always is the whole organism petrified, the whole bone structure. There is actually a T-Rex thigh that was discovered, and it was dug up, and everyone was excited, and someone broke the thigh, which is a big no-no, but it happened. And they actually discovered some soft tissue inside the thigh bone itself. Now, before you start getting excited and start thinking Jurassic Park, we'll start taking the blood out and start amplifying the DNA and cloning dinosaurs. DNA doesn't last that long. It lasts about a couple thousand years, and then poof, it's gone. It erodes away. It's not a very robust molecule outside of living cells. So the fact the organism was dead meant the DNA was going to fall apart through time. And at least 65 million years ago, is too long of a time for the DNA to last. So how old are fossils? So there are 2 ways that we age fossils. One is we call relative dating, and this is based upon the principle of superposition, the idea that lower layers of the Earth are older than top layers. But this just gives you a chronological order. So for example, we know the dinosaurs came before humans. We know that fish came before all the other tetrapods. Fish led the tetrapods, including us, for a matter of fact. And we know this based upon the layers of the Earth and how the organisms are laid out. But it doesn't tell us when the events occurred. Now, there are lots of attempts to estimate this, especially in the 19th century. There are lots of thought processes to try to estimate how old the layers were and everything. But they're almost always off in their own right. So relative dating gives you a chronological order, and that's all that it does. Absolute dating is based upon radiometric dating. This gives you an absolute date itself. So radiometric dating gives an actual time, and it's based upon the predictable pattern of radioactive isotopes transforming from one element to another element. We discussed this earlier in the semester when we talked about the chemistry of life, if I remember correctly. But what it comes down to, if you remember what an isotope is an element with a different amount of neutrons and protons. For example, carbon-14 has 8 neutrons, 6 protons. Oxygen-18 has 8 protons, 10 neutrons. Okay. Now, some elements are unstable when they're in isotopes. So oxygen comes in 3 isotopes. It's oxygen-16, 8 protons, 8 neutrons. Oxygen-17, 8 protons, 9 neutrons. And oxygen-18, 8 protons, 10 neutrons. But those are stable. They're good, and they stay that way for as long as can be expected and all. But some elements like carbon or nitrogen. So nitrogen up in the atmosphere, it's generally nitrogen-14. That's 7 protons, 7 neutrons. And what happens in the upper atmosphere is it gets bombarded by cosmic rays. And when it gets bombarded by cosmic rays, some of those cosmic rays convert a proton into a neutron. So you go from being nitrogen-14 with 7 protons, 7 neutrons, to being carbon-14, 6 protons, 8 neutrons. Okay? Now there's a steady amount of that in the atmosphere. And what happens is that carbon-14, it doesn't want to stay carbon-14. It want to convert back to nitrogen-14. So every 5,300 some years, half of that carbon-14 that was produced that 1 year will convert back to nitrogen-14. And then another 5,300 years, half of that will convert back to nitrogen-14. Now, carbon-14 dating is good for up to 40,000, some say up to 60,000 years ago, ages. So it works well for things that basically are within the human timeline since humans began to radiate out of Africa, which is roughly about 50,000 years ago. Before that, we were isolated to Africa as a species. If you want to go further back, there are other elements, such as potassium, argon. Some versions of potassium will convert to argon. But these work better for 100 million year and billion year range is what they work best for. There's a version of uranium that changes to lead over time. That's also in the billion year range. It's uranium to lead, which led us to the actual age of the Earth itself. So this gives you an actual date with an error range, mind you, but the error range is highly accurate, and the elements themselves are very good for determining dates. Now, the way carbon-14 dating works best is with terrestrial plants. Terrestrial plants sit there and they take in CO2 and breathe out O2, and when they bring in the CO2, they incorporate it as part of their structure. So plants have a carbon-14 signature that matches the atmosphere. But when they die, they stop breathing. Their stomata are no longer functioning. They're no longer processing carbon. So the amount of C14 stays the same within their structure. And through time, that C14 will convert to nitrogen. It makes it an excellent tool for determining dates. So if we find a fossil laying around in the soil, we can date it via carbon dating if it's a recent fossilization event that occurs. And we use other elements for other structures. So biogeography, this is the study of the geographical distribution of organisms. And what this is based upon is the idea that organisms spread out through time. And depending upon the type of the organism, some organisms spread out more readily than others do. Some are what we call colonizers. These are organisms which make it first to new places or places that open up niches, and niches of jobs. So like, for example, when Krakatoa blew up is a big volcano, it denuded the island of life because basically it blew up the entire island. And we got to witness the first organisms that colonized these places. And afterwards, later on, you get the more hardier species which land there, and they begin to take over. For a long time, we didn't quite understand the distribution of organisms. There was a lot of confusion. For example, there were fossilized forms of organisms stretched from, say, South America to Africa to Antarctica, and we couldn't figure out how that worked. It made no sense to us whatsoever because there was huge oceans in between these organisms. But the theory of plate tectonics answered that. This is a theory that states that the surface of the Earth is constantly changing. It's being pushed around and everything. And at one point, all the continents were together in a supercontinent called Pangaea, and this happened at least one other time in the past, possibly another time as well. And at one time, there was a continent called Gondwanaland, and that consisted of South America, Africa, Australia, Antarctica, Madagascar, India, New Zealand, and I'm not sure Australia. I'm not sure if I said that or not, but all these were combined together. So this helped explain the distribution of the fossil record and why it was so odd, why organisms appeared to be related to one another, even though there were thousands of miles of oceans separating them. And this fit in with the geological record and with the biological record of organisms. So the theory of island biogeography, this is a a theory which describes islands and how they have their unique biogeography and their unique organisms. Islands are famous for this. Lots of islands have their own sort of species on it. Islands right next to each other are often their own species. When it comes down to it, is reptiles and arthropods, insect spiders, etc., and birds are the most successful animals that colonize islands. Arthropods because they're easily blown away in the wind. Spiders are particularly good at it. They often stretch out a stretch of silk, and it gets caught in the wind, and they'll be blown away with it. And they'll use this to sort of transport themselves away from their parental insect spiders in a way, not insect spiders, to further move themselves apart so they're not competing with them. Birds are obviously easy enough to describe. They just fly places. Often they get caught in hurricanes too, and they'll be deposited in new places. Reptiles make it very well because they have a tough skin. They don't require a lot of water. In the pirates and the other ships that come to the Galapagos Islands, they would come and take tortoises from the islands because it was a great fresh source of meat, and the tortoises required very little food and very little water for them to survive. So they could take these tortoises on their ships and just wait until they needed them to consume them. So reptiles, arthropods, birds, very good. Mammals, amphibians have a rough time. Mammals require too much energy, too much food, and too much water. Mammals caught at sea have a tough time with it. Not that mammals don't make it to islands, but they generally make it to islands closer to continents, and they generally have a rougher time with it. And amphibians have a rough time too because they have thin skins, and the salt water damages their bodies quite easily. So often what happens on these islands is you have what we call adaptive radiation. And adaptive radiation is an event where evolution appears to occur quicker than most other situations. And the reason why it appears quicker is because when, say, like a bird lands on an island like the Galapagos, a mockingbird, let's say, and let's say the populations of these mockingbirds get separated into several different populations among several different islands. Well, the birds that land on these islands, they can each take new niches that they couldn't take. So if they came from South America already has a complete assemblage of birds, and these organisms that the mockingbird may want to enter their niche would be unable to compete with it because those birds were already established. When they land on an island, now they can change unhindered because there's no one else they're competing with. So when they change, they don't have to be better than anyone else. They just have to change through time, and then they'll take over a new niche. So what happens is you have a lot of speciation events occur on islands, and this is one of the reasons why islands have their unique biota. And another way to look at adaptive radiation is to think about playing a sport. And I always bring up basketball because that was always my favorite sport. But when you go out and you shoot hoops by yourself, it doesn't matter how good you are or how bad you are. You can shoot, and you can shoot for as long as you want, and you get better and better and better. But if you go out and shoot basketball for the first time, and someone's out there, and I don't mean to sound like too fanciful, but let's say like LeBron James is out there, or Kobe Bryant, or Dwayne Wade, or any the NBA players, you're not going to do that well. You're gonna be squished. Basically, is what it comes down to. It's the term always used. You're gonna be squished. You're not going to do well. You're not going to get a point, most likely, unless they're being particularly nice to you, I guess, or something. But anyway, what it comes down to is when you're competing against someone who's already established, already that good, you're not going to do that well. But if you get to compete by yourself, alone, with no one else to compete with, then you're allowed the time to be able to adapt to that particular niche. Okay, so now we're going to talk about anatomical structures, and I just want to mention these three. We're going to go through these with these three different slides. But there are three terms here: homologous structures, vestigial structures, and analogous structures. And these deal with the anatomical patterns that organisms have that are similar, often are more closely related to one another. So homologous structures. These are anatomical structures which share the same evolutionary origin. They have the same genetics involved with their development, meaning they have the same genes, and they have the same embryonic development. So a good example of homologous structures are the forelimbs of tetrapods. Those are the forelimbs, F-O-U-R, not just F-O-R-E-L-I-M-B-S, of the forelimbs of the arms and the legs, or the forelegs of a horse. Amphibians, reptiles, birds, dinosaurs, and mammals, they all have the same structure, the same genetic origin, and the same embryological origin, meaning it develops in the same pattern. And actually, there's a neat thing. There's one bone, two bone, and a blob of bones for each of the legs and arms, meaning you have one bone in your upper arm, two bones in your forearm, and then you have a set of bones for your fingers and your hand and your wrist. This is a common pattern. And if you watch the embryology of these organisms, some organisms lose part of this as they develop their embryology through the development in the womb or in the egg if it's a egg-laying animal. Such as horses, they have one primary toe. If they lose a lot of this toe, the other toes become what's called vestigial. We'll get into that in just a second here. Another good example of a homologous structure are the gills in fish. So gills, jawless fish have 7 gills. They don't have jaws like we do and like the jawed fish do or any of the other vertebrates. Jawless fish have 7 gill structures, and those are used for respiratory structures. And what happened is a regulatory gene mutated, and eventually fish had jaws. They lost the gill structure. Now, if you trace our embryology back, we have the same gill structures in our embryogenesis when we're developing. But our first gill, if you want to call it that, it's essentially what it is, becomes our jaw structure. Okay? And we can trace this pattern throughout all of the tetrapods and the fish based upon our evolutionary history. It all comes from the same genes, it's just reorganized. Matter of fact, vertebrates as a whole, vertebrates include fish, amphibians, reptiles, mammals, birds, dinosaurs. They're all pretty boring. They're all based upon the same anatomical structure. They all have a backbone, a cranium. When you get in the tetrapods, you have 4 limbs and a pelvic structure and a pectoral structure. So it's all the same pattern. It's all because we share the same origins. So vestigial organs, these are organs which used to have a biological function but no longer have a biological function. And this fits in with evolution because it would make sense. If evolution were occurring, you'd expect to find some organs which are no longer useful in organisms that are still there because they used to have use. And it takes time to lose them. So some good examples are the piloerector muscles in our skin. These raise the hairs on your body. For example, the hairs in your arm or the back of your neck. You may have been at a concert and particularly got excited when a particular line got sung and you felt the hairs in the back of your neck stand up. Or you might have been in a situation which was very scary, maybe at a movie or maybe something actually was happening to you and you felt the hairs in your body stand up. So the reason why this happens is back in the day we used to have a lot more hair in our bodies. And what happened was when the hair stood up, it was a way to poof up, to make you look bigger. So that way you could intimidate something that was coming at you. Your cats and dogs do this. If you ever notice your cat and dog, whenever they get particularly excited or when they see something that really scares them, their hair stands up. It's to make them look bigger and all. There's another reason for this too. One is body warmth. Our hairs stand up on our body. Those are piloerector muscles that we call goosebumps when it's cold out. And that's because we used to have a lot more hair in our bodies, and the hairs would rise up, trapping the air against our skin, and as our skin radiated warmth out of it, it would warm that air around our body, and it would keep our bodies warmer for longer periods of time. So that's one of the vestigial organs that we have. Whales and snakes still have pelvic structures from when they had 4 limbs. Whales come from organisms which are very closely related to hippopotamus, and they used to have 4 limbs. And the whales today, they still have the pelvic structure. That's the portion which connects to your legs. They still have that in the hind portion of their body because that's where they come from. If evolution hadn't occurred and these organisms just appeared in the water, you wouldn't expect them to have these structures because there's no need for them to have it. But because they came from an organism that was terrestrially based, they still have those structures and those genes are still active during their development. So, vestigial structures tell us evolutionary history and it can unite us with previous organs. So does homologous structures. Analogous structures are a little bit iffy in a way, and they can throw scientists off. And these are structures which appear the same, but come from different evolutionary origins, different genetic structures, okay, different genes, okay. And this often leads to what we call convergent evolution. This is where a particular structure is highly successful, and different organisms evolve this structure separately from one another. So a good example of this is the streamlined shape of the marine mammals and the fish. Okay, the streamlined shape is very good for swimming in water. You can attain higher speeds with it and all. Now, the dolphin streamlined structure is a bit different from that of sharks, but they still have the streamlined structure. If you notice, a shark's tail, caudal fin, moves from side to side versus a dolphin's caudal fin moves up and down. Okay, it's a different way that they approach the motion in water. And some structures are both homologous and analogous in a way. When you look at, say, the wings of a bat, the wings of a bird, from the limb point perspective, they're homologous. They both come from the same structures that our arms come from, that of a bird's wing and a bat's wing. But the wings themselves evolve separately. The actual structure of the wings. A bat's wing is made of, its 5 fingers are very extended, and they encapsulate basically a skin, which makes a wing structure, versus birds. They lose some fingers through development, and they basically have a couple fingers which are really strong, and they have feathers that come off of it, which allows them to fly and keep their bodies warm too. So some structures are a little crazy one way or the other. And in the past, I could see where bats were classified with birds because they both fly. And that would have been a common mistake done by previous populations in our history of a species. But of course, bats are mammals just like we are. We have the same evolutionary origin from that perspective and that point. So embryonic development. Evolution can be traced through embryonic development of organisms. Organisms that are more closely related to each other have the same similar embryonic development. Haeckel, who was a researcher in the 19th century, he very sadly threw the research off by when he drew pictures of the embryogenesis of organisms. He didn't draw them too accurately. He made them look more like similar to each other than what they were, which is a shame because it wasn't necessary, it wasn't needed. But and to tell you the truth, I don't know how much of it was just the lack of his ability to see. Maybe his eyes weren't as good as what they were thought of as being. Maybe his microscopes weren't all that good. So there's a lot of possibilities there. It may have not been for deceitful reasons. But when it comes down to it, you trace our origins, our evolution, and you trace our embryonic development. There are phases where we do look more like a fish. There are phases during our embryogenesis. And the more closely related you are to an organism, the more you look like them. Chimpanzees and humans look a lot alike. When chimpanzees are born, they look more human than chimpanzee to some degree. Their facial structure is much flatter, like ours is. It's not until as they develop that they develop this stronger jaw structure, which juts out from underneath their cranium. An important gene is homeotic genes. These are genes which affect other genes. Is what they do, and they control the development of organisms. So chimpanzees and humans are considered to be very genetically similar to one another. But a few changes in homeotic genes, which cause a cascading effect of other genes during our development, during our embryogenesis, change the structures of our body. The same thing as homeotic genes, which led to the gill structures that we have during our embryogenesis become different things. Okay, like I said before, the first gill structure becomes our jaws. Other gill structures become our inner ear, our trachea, other parts of our body. Okay. So Ernst Haeckel, I forgot that I had the slide come up after it, but I think it's important to point it out because he still talked about, not by biology classes mainly, as much as it is by people opposed to the theory of evolution. He's brought up as thought of as being like his research is still used and everything in textbooks and things of that nature, and it isn't. His data is no longer used, has been used for decades, but it's another thing altogether. It's unfortunate because it puts a negative connotation on embryogenesis, the study of embryology. It also gives a bad sign for science, but science is like everything else. Science is a tool. Scientists are the people, and some scientists make mistakes. Some make it by accident, some do it on purpose. So, but his research has been discarded and replaced with more accurate research ever since. So, comparative molecular biology. This is the more exciting and more recent trend that we've been doing for the last 25, 35 years. We've been doing research more in DNA, RNA, and proteins of organisms because this gives us a more accurate approach to it. So it nullifies the whole analogous structure because if two organisms have structures which are similar to one another and may throw a scientist off, if they don't share the same origins, then then there'll be different genes involved with those structural components of the organisms. So we can trace back our mammalian history by the genes. We can trace our ancestry with fish based upon the genes, and amphibians and reptiles and such. Okay, so organisms with similar patterns in the DNA, RNA, and proteins are more closely related to one another than others are. Okay, cytochrome C gene is featured in your textbook in Figure 12.19. That's just one of the many genes that we use to determine phylogenies. When I was an graduate school, sorry, I was working with a gene called the cytochrome B gene, which is a gene found in the mitochondria of organisms. And we were trying to isolate that gene, sequence it, in order to determine how a group of fish from the Indo-Pacific region were related to one another. So here are some examples. Humans and chimps. Human chimps are very closely related to each other. There's another group called bonobos, which are highly related to chimpanzees. When it comes down to it, bonobos and chimpanzees radiated from one another after humans and an ancestor that was also connected with chimpanzees and bonobos radiated from one another. But when it comes down to it, is all the apes have 48 chromosomes and humans have 46. And there's a big debate about this. How could this be? How could organisms that are so genetically related to each other, how could one of them be missing an entire chromosome and yet then be so similar to one another? So this is a problem, and this is the dilemma within science. So what the evolutionary theory predicted is that what we would find in our genome is a chromosome that is coupled together. And the way you could tell whether a chromosome was basically two chromosomes coupled together was if you found telomere DNA in the center and two centromeres. I don't know if you remember this, but the telomere is the end part of a chromosome, which is just junk information. And it's there to act as a fuse for the life cycle of that particular chromosome. Well, so we started scanning through the chromosomes of the human genome. And what we discovered was the chromosome that we've designated chromosome number 2 has a centromere in the middle of the chromosome and a telomere in the middle of the chromosome and two centromeres, although one of the centromeres has become dysfunctional. And on top of that, the genes in this chromosome, that's two coupled together, matches the two chromosomes that are found in the chimpanzee. Okay? And this isn't the only time this happened. It looks like it happened between donkeys and horses. One of them, I can't remember which, has an extra pair of chromosomes, the other one doesn't have. So this isn't uncommon amongst organisms to have their chromosomes fused together. So there's another piece of evidence which suggests that humans and the other apes share a common ancestor, and that is the vitamin C gene. So most organisms produce their own vitamin C, but we don't. It was a big thing when shipping was first getting into being and all and everything. Sailors often got scurvy, which is the result of lack of vitamin C. But it turns out that back in the history of the primates, some of the primates that led to the apes lost the ability to produce their own vitamin C, and that's because they're eating lots of fruits. And when you're eating fruits, you're getting lots of vitamin C, so that gene is no longer necessary. And what happens is, the gene is no longer necessary. If it mutates, it's not a problem for the organisms. And it began to mutate with that common ancestor. And with that mutation, it led to a vitamin C gene that's no longer functional. Now on top of that, what would you expect to have happen if that gene became dysfunctional in the past? You'd expect all the organisms that preceded it, come after it, to have the same mutations. If chimpanzees, bonobos, gorillas, orangutans, and humans all didn't share a common ancestor, and all didn't have a functional vitamin C gene, which is just as likely, you'd expect them to have different mutations. You'd expect different things to have occurred within the genes to make it dysfunctional. Well, it turns out we have the same 6 mutations that lead to the dysfunctionality of that gene between these various groups of organisms. And it's one of the smoking guns that supports the whole theory that chimpanzees, bonobos, gorillas, and orangutans, and humans all share a common ancestor. So the evolutionary tree of the great apes, this is just demonstrating our history. You have the orangutan and the gorilla. The gorilla was at one time considered the closest living relative to humans. I think Alfred Russel Wallace, the other person who came up with the theory of natural selection alongside that of Darwin, he liked the idea of the orangutan being closely related to us. Wasn't until later on we discovered the chimpanzee and bonobo are more closely related to us than the gorillas or the orangutans. Matter of fact, chimpanzees and bonobos are more like us than they are any of the other primates. And the same thing can be said about us and them. So there's another thing which we can use, and this has become a recent thing. Matter of fact, when I was in graduate school, this was awfully sketchy, but this has become more of a thing through time. It's what we call the molecular clock. And the molecular clock is a method of determining when two groups of organisms diverge from one another. Okay? So 2 pieces of knowledge have to be in. One is the rate of mutation for a particular gene. Not all genes mutate at the same rate. And the reason for that is because some genes are more sensitive than others are. So if they mutate, it's a bigger effect. Some genes can mutate more frequently. It doesn't have much of an effect on the organism. Okay? Mutations which exist between the 2 individual groups of organisms must be measured. But based upon this, if you have the knowledge met, you can come up with a general idea of when 2 organisms separated from each other based upon the amount of mutations that have occurred between the 2 organisms. So this is one of the methods we use to determine the evolutionary history of humans. Now, the history of humans and our ancestors, chimpanzees and bonobos, have been pushed back further. I think it's like 8 million years now. Before that was 6 million years. And that's because more and more evidence came in. As evidence comes in, we refine our results, and things can move further away or closer to it or whatever, but that's what we do as scientists. So here's a little bit. So Darwin's finches, this is something which people often think Darwin studied. He didn't really study the finches. But there are 2 ecologists, Peter and Rosemary Grant, from Princeton University, and they've been on the Galapagos Islands for something like 40 years. They've been doing research on the Darwin finches. And the amazing thing about that is they've seen evolution in action. They've seen finches speciate out from one another, where a population of finches could breed with one another, and it got to a point where it split into 2 subpopulations, where those subpopulations could no longer breed with one another. That is a speciation event. That is a macroevolutionary event. That is evolution in action that they got to witness. Now, this is the only example of this that's occurred. This has occurred in mice. This has occurred in bacteria. The smaller organisms are much easier to discover this in because they procreate so quickly and so rapidly, and their generation time is so quick as well. Humans are very difficult because we have such a long time period between generations. Same thing as whales. The bigger the animal, generally, the longer the generation patterns are between them. But here's an example of evolution in action, of speciation in action, of Darwin's theory in action, and it supports the whole premise of evolution.